All right, good. Let's come back here to chemical composition, right? Good. Chemical composition. Now, in your chemical composition here, first thing we have there is the basal or simple media. Now, your basal or simple media is simply like your plain nutrients media. Two things are basically present in your basal or simple media. It's just like where you want to make something and then you just have the plain side of it, the something you want to make. I get me now. You just have maybe I want to make you can make sandwich in different ways where you want to put all those plenty extra things, but we know that the basic sandwich is just your bread and your butter. Then if you want to add sardine, you add it. If you want to add those other things, they add it. Those are nutritional varieties. Same thing here. So that basic major component where you are just having the bread and butter is what we're talking about here in basal and simple media. So the two major things you want to have in your basic your basal or your simple media is just I'm coming here, basal or simple media. You just want to have your beef extract, or you can call it your meat extract, which is usually obtained. Your meat extract, that is from meat. You extracted the fluid or a liquid from meat. Are you there now? So you want to have just your meat extract, and then you want to have peptone water. So meat extract plus peptone water is the major component of your basal or simple culture media. Don't forget that meat extract, you get it from lean beef. Basically, you get it from lean beef. So once you have your meat extract and then you have peptone water mixed together, you already have a simple what do you call it? You already have a simple media or a basal media. Now, what's the importance or the use of a basal media? Now, note it does not have a lot of nutrients in it. It's just basic nutrients, right? So that is why this kind of media is usually only going to be useful in microbes we refer to as non-fastidious microbes. So this is found useful in non-fastidious microbes. Now, when you hear us use the word non-fastidious microbe, a non-fastidious microbe is a microbe that do not have large nutrient requirements for its growth. So we just need simple or basic nutrient requirement and the organism is growing. So you get that now. So you just have your normal meat extract and peptone water, and that's enough nutrient requirement for the bacteria or the microbe. That means that microbe is non-fastidious and then it's useful there. That means it can be grown in a basal or simple culture media. Very, very simple. Now, let's quickly analyze something. If you add glucose, for instance, if you add glucose to this mixture here, what you formed now, what you had before is called a nutrient. It's called a nutrient broth. Are you there now? Nutrient broth. Why am I using broth? Because it's still going to be in liquid. When you add glucose to it, the resulting component you now have is now referred to as a glucose broth. A glucose broth. Now, if I now even go further and add agar, which I told you agar is the what? Is a sulfate polymer of D-galactose, 3,6-anhydro-L-galactose, and D-glucuronic acid. If you just add that into this mixture of meat extract and peptone water, what you will now be having there will now be called a nutrient. Look at the difference. A nutrient agar. So if they ask you to differentiate between a nutrient broth and a nutrient agar, a nutrient broth just contains meat extract and peptone water, but a nutrient agar has meat extract, peptone water, and at least 1.5% of what? Agar in it, and that gives you that. Remember, non-fastidious microbes. Let's go to the solid culture media now. The solid culture media. So for your solid culture media now, okay, we've done this already. That's what we talked about here. Solid culture media. Sorry, we are already at chemical composition. We've talked about this now. That's the basal simple one. Now, what about the complex media? Now, your complex media Pay attention to that. Your complex media are used in growing fastidious microbes. Fastidious microbes now are microbes that have what? That have certain or huge nutrients requirements for growth. So it can't just be meat extract and peptone now. You will need extra kind of what? Nutrients. So your complex media is a media containing varying is the key word there, because that's what makes the difference between complex media and definite media. Complex media, the nutrients are varying. Definite media, the components are fixed. I have this percent of this and this percent of this and this percent of this fixed. But in complex media, you have varying varying composition of nutrients. Are you there now? Within the media, you have varying composition of nutrients within the media. What are the major examples of your complex media? I told you they are used in growing fastidious microbes. So what are the major examples of your complex media? This is where you learn about McConkey media. Please take note. The McConkey media. And we are going to look at what are contained in this McConkey media and all. So we have the McConkey media in this. You also want to talk about your triptych media. So we have the McConkey media and we have the triptych media. So these two media here are major examples of your complex media. We have other medias. We have the insane methylene blue, what they call it, agar media. You also have your endo agar media. All those things are examples of complex media. But for the JUPEB, for the sake of the JUPEB lecture, you just want to learn. We're going to be exploring the McConkey media and the triptych media. Now, the thing there now is, we said they have different ingredients, different nutrients present in them. As a JUPEB student, you need to know the components of the McConkey media. You need to know the components of the triptych media. So what are the components of the McConkey media here? So we're going to bring that here now. I'm going to bring that here. Components of McConkey media. So what are the components of the McConkey media? So inside the McConkey media, you're going to find your pancreatic digest. And there are two major pancreatic digests you are going to be finding in this. You are going to be finding the pancreatic digest of gelatin, and you find the one of casein. So pancreatic digest of gelatin and pancreatic digest of casein. Casein is a pancreatic digest that you actually get, is a protein usually associated with milk. So just know that. Then you have your pepsin digest. That is things digested by pepsin juice, right? Now the next things I want to be writing are very key to McConkey, because we are going to use that to explain certain things about your differential and your selective media. So what are those things? This McConkey also has lactose in it. Remember, that's why it's a complex media. It has so many things. Lactose in it. It also has bile salts in it. It has bile salts. It also has agar in it. So we know that it is going to be solidified, right? It has agar in it. One thing again that this guy has is your crystal violet dye. Crystal violet dye. It has it in it, yes. It also has neutral red salt. Oh, no problem. It also has sodium chloride as well. I'll just put that here, NaCl, and your neutral red salt. So all these things here are the major component of your McConkey media. No glucose is in McConkey media. You find glucose in triptych media. Triptych media basically just contain, let's just put it here. Your triptych media, let me write it here. Your triptych media, sometimes you see some people call it both. So that one just contains, basically, you'll find your pepsin digest also there. You find your pepsin digest, but most especially you'll find your tryptone, which is why the name triptych media, and then you'll find your glucose. Yes, So basically, this is the major component of that. So sometimes this triactic media, you see them call it triactic soil. Let me just call it triactic soil media. So this triactic soil media can be triactic soil broth or triactic soil agar. If it is a triactic soil agar, automatically you are adding what to the component agar. So that's that for the complex media. So the complex media are used in growing fastidious kind of bacteria that have a large amount of nutrients requirements. So the bacteria might need lactose, it might need bile salts, it might need agar, it might need crystal violet dye, it might need pancreatic digest, it might need pepsin, whether pancreatic digest, gelatin or casein, all of them are present within the McConkey media, and that is why this is basically used for such kind of bacteria that need a lot of nutrients. So there is high tendency nutrient to be amongst this. Now, in your definite or synthetic media, not much to say here now. Let me bring out one difference. You see, this is your complex media, right? You get most of its extract, all this McConkey and all this triactic soil, you get most of its extract from 3 major sources. Get me now? From 3 major sources. What are the 3 major sources you can get those guys from? That's these nutrients that you find in complex media. You either find those things in animal tissues, so you can say extract of animal tissues. You can find them from plant tissues, extract from plant tissues, and you can also find them extracted from yeast. Yes, so you can see that there you have them gotten from natural sources. For example, that lactose, we know that you get lactose from animal tissues. You understand what I'm saying? Bile is also from animals. Also, you can find what they call it, crystal violet and salt from also plants and animals, and so on and so forth. That's what I'm trying to state there. They're usually from natural sources. But the definite or synthetic media, you do not have whether plants, animals, or yeast extracts. None of them are present there. This guy has also a definite proportion of nutrients within its within its media. So it is only used to grow a specific type of microbe. You get now? So definite, the configuration of its component has been made so that it will only suit the growth of a specific type of bacterial population. So that is that for that. Now we're going to be discussing the classification based on functions. We have the selective media, you have the differential media, you have the transport media, you have the enriched media. So basically, what is selective media? What is the selective media? Just get me, it's very, very simple, I promise. Now, your selective media here now, it is a media that has the ability, okay, let me put it this way. It's a media that contains ingredient that has the ability to inhibit the growth of some microbes and support the growth of another type of microbe. So if you have a culture now where there is a number of different populations of bacteria, I get me now, the selective media can help in the inhibition of the growth of one kind of bacteria while it is enhancing the growth of another, so that you can, in that kind of case now, if I have both gram positive and gram negative bacteria in one mixture or in one culture, are you there now? There are certain kind of there's certain kind of mediums, that's culture mediums, that could help or inhibit the growth of gram positives while it is enhancing the growth of gram negative. That is a selective media. So a selective media is a media whose nutrients can inhibit a specific strain of microbes while enhancing growth of other microbes. A good example of a selective media is your what? A good example of selective media here is the McConkey media. I said it there that its application applies here. You have also the enzyme methylene blue agar media. You have that there. So 2 major ones I've given you there. So how does this work? Let me just use the McConkey as an example. Now, McConkey as a selective media, there are 2 major things or 2 major nutrients that's present in McConkey that makes it a selective media. And what are those 2? We have the bile salts and we have the crystal violet dye. How? Now, gram-negative bacteria, like we said earlier when we were looking at morphology and structure of our microbial cell, we know that gram-positive bacteria have thick peptidoglycan cell wall, right? While your gram-negative lack peptidoglycan on their cell wall, or they have a thin one. Good. Another thing again we know about them is that your gram-positive bacteria do not have an outer membrane, right? While your gram-negative has an outer membrane. Good. Now, whenever you are using the selective media, that is McConkey for instance, that BIO002 tends to alter the integrity of the cell wall of your gram positive. Why? Because there's no outer cell wall, so it easily disrupts the peptidoglycan and affect or cause perforations to the inner cell wall. No outer cell wall, so that means there's no protection. So direct attack on the inner cell wall will cause perforation, and that will either lead to death or inhibition of the growth of those kinds of bacteria. So you can see how BIO002 in McConkey has been able to select what they call it gram-negative over gram-positive. How come? Because the gram-positive do not have an outer membrane, so there is direct attack or there is direct susceptibility to inhibition of the inner membrane in gram-positive. So what about crystal violet dye? Your crystal violet dye there usually will alter the peptidoglycan configuration of your gram-positive and also lead to inhibition. So you can see BIO002 and crystal violet dye, they select gram-negatives over gram-positive. When we say select gram-negative, that means they will usually support the growth of gram-negative while they are inhibiting the growth of gram-positive due to presence of BIO002 and crystal violet dye that can lead attack or destroy the cell wall of the gram-positive due to the absence of outer cell wall. You know, gram-negative has outer cell wall, so before you even start dealing with the inner cell wall, every other thing there has been sorted out. You understand? The protection is there. All right. Then we'll look at the differential one now. The differential media. Now, your differential media, right? This guy here has the ability. Now, it is not inhibiting. It is not inhibiting the growth of another one. Rather, it has nutrients that has the ability for the scientists to differentiate between different strains of microbes in the culture. So we have certain ingredients that we can look at and be like, oh, these ones are this set of bacteria, these ones are this set of bacteria. For instance, now McConkey is also a differential one. You can see that now. So McConkey media is selective and is differential. But what makes it selective is BIO002 and crystal violet. But what makes it differential is the presence of lactose. You can see that now. It is the presence of lactose. How? We know that some bacteria can ferment lactose, while some cannot ferment lactose. I don't know. Those that can ferment lactose will ferment lactose to turn it into pink. Those that cannot ferment lactose, it will form final solution color of what's colorless. So it means that if I have a media like this, for instance, now, if I have a media like this, for instance, maybe this is a media, this is a petri dish, and inside my petri dish here now, I put in agar solution, and maybe the agar I'm using is McConkey. Maybe I'm using McConkey here. If I put the McConkey and I notice that I'm seeing pink colorations at maybe all this side here, pink colorations here, and around all this other side here, I'm seeing colorless. I'm seeing colorless holes. Are you getting now? You know that it's a solid brute. So normally it's solid, but if I start seeing some hole, like colorless hole, on some regions, and I'm seeing red patches on all these red patches, I can easily know that the bacteria here are bacteria that have the ability to do what? Ferment the lactose that is in the agar. Why these ones here that are colorless is as a result of bacteria in this place that are non-fermenting bacteria. So very, very easy. Very easy. So that's the differential kind of medium. Another important example of a differential medium. Now, I told you that McConkey is a differential medium. McConkey is a differential medium. Another differential medium is blood agar. Because some bacteria have the ability to hemolyze blood cells, while some do not have the ability to hemolyze blood cells. Now, if this was a blood culture, for instance, you have a blood sample, a blood agar, and on this blood agar here now, you're not seeing some parts in the blood agar that is patches of empty spaces, in the blood agar. This explains to you that all these regions that they are patches, the bacteria that are present in these regions that they are patches are what? Are the hemolytic bacteria. Why are there holes in this place? That means those bacteria that have the ability to hemolyze red blood cells, that's to destroy or to kill red blood cells, are present in these regions. Are we together now? They are present in these regions. So automatically there, you can come to a conclusion that the bacteria present in all these places that are holes are hemolytic bacteria, while other bacteria in this culture will be what? Non-hemolyzing bacteria. So you're seeing about differentiating hemolyzing from non-hemolyzing, fermenting from non-fermenting using McConkey. Those are differential media. So in differential media just helps you to use certain processes of those bacteria to differentiate between the cultures or the bacterial species present within that microbial culture. Now, what about a transport media? What's a transport media? A transport media. Now, you know, microbes are very, very sensitive. They are very sensitive, especially to motion. And it turns out that you can't just keep them in the laboratory just anyhow. There are going to be sometimes you want to transport them from one point in the laboratory to another point in the laboratory. That period where you are transporting bacterial cultures from one point in the laboratory to another point in the laboratory can lead to destabilization of the bacterial cell. Yes, and that is not what we want, because we said one of the goals is to preserve them. Why? Because we might want to use them for certain kind of research. So in moving bacterial cells from one point to another, you will need a transport media. And the major most common transport media that we have that does that is your what? Is your SWAT media. SWAT media. So that is it. So I call it a SWAT agar or SWAT both, depending on the nature of state of the medium. So transport mediums are usually useful whenever movement of culture from one point to another is going to be happening, or from one laboratory to another laboratory. So that's for that. What about your final one, which is the enriched media? The enriched media. So this one is actually very, very simple. It's very, very simple. The enriched media basically is just like when you have your normal plain media and you add extra nutrients. So an enriched media just have extra nutrients. Why do you want it to have extra nutrients? That means are we supporting non-fastidious or fastidious? It's because we want to support fastidious. So, you know, maybe before the amount of nutrients there were limited, now you just give it a blood bath, or you added yolk to it. So the common things they usually add in enrichment media, that's to the normal media that was existing before, is either they add blood or you add yolk. So anything that has source of nutrient, extra source of nutrient. So an enrichment media is just a media that has been treated with extra nutrient source. Extra nutrient source. So two major ones are blood and yolk that you usually add into it. So with what has been discussed right now on the board, we have come to the end of culturing. This describes everything under culture media. We have not come to the end of culture, we have come to the end of culture media. So we have been able to explain the different types of culture media, and this is everything you need to know about different types of culture media as an A-level student. In our next lecture, we'll be describing different types of culture. There's a difference between different types of culture media, there's a difference between different kinds of culture. Good. I remain my humble self, and this is Henry's Impact International Academy.